Right, next up, we have Sarah Middleton from the University of Pennsylvania. Her field is in genomics and computational biology, and her advisor is Junyoung Kim. Her practicum was at Lawrence Berkeley National Laboratory in 2014.
Hello, everyone. So today I'd like to talk about a method I've been working on for finding secondary structure motifs in RNA sequences. But before I get into the computational details of the method, I wanted to take a step back and talk a little bit about the biological question that first got me interested in this problem. So one of the things I study is neurons, and in particular trying to understand the molecular underpinnings of some of the interesting things that neurons do. One of these things is a process called long-term potentiation, or LTP. This is basically the process by which over time a neuron alters itself to become more sensitive to input. And it's thought that this may be one of the important processes underlying the larger process of learning and memory in the brain. So the way this basically works is that when a synapse is activated, it undergoes a process of remodeling where it'll add new membrane channels, receptors, or grow in size. And the result of this is that it then becomes more sensitive to future input. But one thing that was noticed a few decades ago is that for this process of long-term potentiation to last, it's not sufficient for the synapse to simply recruit existing proteins from the surroundings. It actually needs to do new protein synthesis. By that, I mean that it needs to take messenger RNAs and perform translation to create new protein. And we know this because if we inhibit the process of translation, this prevents the cell from having this long-term potentiation last. Usually it can last on the order of hours or even longer. So this presents something of a logistical problem for the cell because, as many of you know, RNA is produced from DNA, which is stored here in the nucleus. So all new copies of RNA that are produced start out here in the cell body. So the cell needs to figure out some way to produce proteins from these RNAs, but make sure that they only get to the synapses that have actually been activated. Because you can imagine if they go everywhere, that's going to undermine the process of long-term potentiation. So the cell basically needs to find a way to get these proteins to the right place only at the right time. So the way that it actually does this is pretty elegant. At least this is one model. The cell will pretty much constantly transport RNA out to the dendrites, just everywhere, but it'll keep the RNAs in a translationally repressed state, so they can't be turned into proteins. Then, only when a synapse is activated will that repression be relieved so that the RNA can be turned into protein. And what's great about this is, well, two things. For one, it keeps the protein relatively constrained to the area that was activated, and it also allows the response to be very rapid. Because everything that's needed is right there. So since this theory was developed, it's been observed that both RNA and also translational machinery, such as ribosomes, have been observed out in the dendrites. This picture shows what's called RNA fish experiment. Basically, in this experiment, we have fluorescent probes that are specific for certain types of RNA. And when we add these to the cell, they'll bind to the RNA and show their physical location. So each of these dots represents a single RNA molecule, and they're shown along the length of a dendrite. So you can see that many of these RNAs are present even out in the distal regions of the dendrites. But what's really interesting is that actually not all or even most RNAs show this pattern. It seems that most RNAs stay relatively restricted to the cell body. So this really raises the question, how is dendritic localization regulated? How does the cell decide which RNAs are localized and when? So what do we know so far? Well, we think that the way that RNAs actually get out into the dendrites is by the use of transporter proteins. The way these proteins work is they're basically kind of like cargo trucks that ship out the RNAs to where they need to go. And the way they recognize which RNAs they need to ship is by recognizing specific motifs that are actually on the RNA itself. These motifs can either be linear, so just a linear sequence of nucleotides, A, U, G, C, or they can actually be structural in nature. So an RNA structure is when the single strand of RNA loops back around and actually base pairs with itself. So A is pairing with U's, G is pairing with C's. And in this case, we think that is actually the structure and not so much the actual underlying nucleotides that is what's recognized by the transporter protein. So it's the presence of these motifs on an RNA that lets the cell recognize which ones should be transported. So clearly, these motifs are pretty important for this interaction between RNA and transporter proteins. So we'd like to kind of figure out what these motifs are. You can imagine that if we had a person that had a neurological disorder and we found out they have a mutation in one of these motifs, that would give us a good hint of what's going wrong. If we knew the identity of all the transporter proteins, there are actually some experiments that we could do that would allow us to identify both which RNAs that protein is binding to and also where it's binding. So that would let us find the motifs. But unfortunately, at the current time, we don't really know the identity of a lot of the transporter proteins. But we do have one piece of information that's helpful. We know that it's probably not a one-to-one relationship between transporter proteins to localized RNAs. And that's because there's only on the order of maybe dozens or less transporter proteins that we think are actually involved in dendritic localization, whereas there are maybe hundreds or thousands of RNAs that are actually localized. So what this means is that a single transporter protein type must recognize multiple RNAs. And that means that those RNAs must share a motif. So we can probably use computation to identify that motif. So there are some really great methods already out there for identifying linear motifs. So not surprisingly, people have already tried to do this. Basically searching all the known localized RNAs, trying to find whatever motifs we can find. But kind of surprisingly, we haven't found anything yet. We found some isolated motifs that are on just one RNA but don't seem to be found on any others, so they can't really be called motifs per se. But really not that much has come up in terms of a unifying motif that would explain localization. And so now one of the theories is that actually it's structures that are playing a big role in localization. Now the problem with structures is that we don't have as good methods for identifying structural motifs. And I'll just give a quick example to show why this is the case. So here are two sequences that form the same structure. But if you line them up and look at how many actual nucleotides they share in common, you see that only these three highlighted ones are in common. So those sequences look completely different, and yet they form the same structure. And that's because it's really just the base pairing that's important. You can swap out an AU base pair for a GC base pair, and usually it won't matter. So this is what makes this problem so difficult. So now this brings me to the main point of my talk, which is how can we find RNA secondary structure motifs. So I'm just going to reframe this problem in a more general sense, and then I'll quickly go over two methods that have been used in the past and why they sort of aren't sufficient for genome-scale datasets. And then I'll talk about the method that I've created that will hopefully help address some of these problems. So our main task is basically if we have a set of sequences of unknown structure, so we don't really know anything about their structure yet, can we predict subregions of those structures, subregions of those sequences that have a common structure or motif? Some complications with this. We can't assume that every sequence will have a motif or that every sequence will share the same motif. And we don't know the boundary of individual motifs ahead of time. So RNA molecules are usually fairly long, and we expect the motif to only be a short part of it. So these things will make this a little bit more difficult, and I'll talk about how we can address them later. So one of the methods that currently has been used is what's called fold and compare. So in this case, you take each individual sequence and you compute the minimum free energy structure for the sequence. And then once you have a structure prediction, you just compare each pair of sequences and compute some kind of structural similarity score. So the advantage of this method is that it's fairly fast, but the disadvantage is that unfortunately, it turns out in real life, RNAs don't actually always take on the minimum free energy structure. So this ends up being not quite accurate enough for our purposes. Another more accurate method is called align and fold. In this case, you take each pair of sequences and you compute an alignment that just try to simultaneously optimize the covariance information and also the free energy structure. Covariance information is basically just looking at conservation of base pairs rather than simply just conservation of nucleotides. And I don't have time to get into the details, but basically it helps you identify what's more likely to be the structure taken on in life because it's the structure that's conserved. So this is better because it's more accurate, but unfortunately each alignment is extremely slow, and so if we have to do n-squared alignments for our input sequences, that just doesn't scale well enough for large-scale data sets. So here, this brings me to our method. So the basic idea here is that instead of comparing the sequences to each other up front, what we're going to do is compare each sequence against a set number of landmark structure models. So these structure models are chosen to just represent a wide variety of structures that are taken on in nature. They're represented by what's called covariance models. These are basically a type of context-free grammar, and they're good at just representing nested structures. And there's already a program that will score a sequence of unknown structure against a model and basically spit out a score that tells you how likely that sequence is to take on that structure. So we can score each sequence against these models. That will give us a vector of scores that we can then use like a feature space to do clustering. And ideally, if this works as intended, we'll have sequences that have a similar structure being placed in a similar area of this feature space, and that will allow us to very easily pick them out as clusters. So just to benchmark this, first we created a synthetic data set. Basically, we generated sequences that were pretty much random, except they are constrained to form one of these three structures. And just to emphasize, these sequences are basically, for a given structure, look essentially random. Like you would not be able to pick out that they are the same structure just based on their sequence alone. When we mapped into the feature space and we measure the distance between each pair, we see that structures that have, sequences that have the same pair, same structure, are closer together in the feature space. So this indicates that we're getting exactly what we wanted here. Sequences, same structure, are mapping together. And in fact, if we do a PCA, you can see that that's exactly what's going on. So now, of course, that's a synthetic data set, and biology is messy, and we can't always rely on synthetic data sets to really reflect reality. So we next tested this on a real data set. We took 20 known structure families, things like tRNAs, well-established structures, filtered out their sequences, just to remove anything with trivial sequence similarity, because we want this to be kind of hard test. Mixed all the sequences together and mapped them to the feature space, and then did an unsupervised clustering. So if we do a low-dimensional projection of the space, we can already see that sequences are clustering by their family. And if we do clustering using hierarchical clustering, we see that the sensitivity for almost all the families is really high. So this is working really well. And also I want to note that the specificity is really good too. So clusters basically contain mostly all one family, and they contain mostly all of the members of that family. So this is exactly what we want to happen. So now you remember I talked about two complications that arise in the real life. So we want to try to address the problem where sequence may have no motifs and maybe kind of just like a noise in the data set, and the fact that we don't know the boundaries of the sequences of structures. So to address this, I created one data set that had added decoys. These are basically just randomly sampled from real RNA, but are not expected to have a structure that's related to the ones that we're trying to target right now. So they're decoys. And then I also created a data set that is basically the same as before, but had a random amount of random sequence on either end of the structure. So that will that's kind of simulating the idea where we don't know where the boundaries are. So on the data set with decoys, we actually do pretty much exactly as well as we did before. So this method works really well for rejecting stuff that isn't actually a motif, that doesn't share commonality with anything else in the data set. However, in the case of the embedded sequences, we have a little bit more trouble. Unfortunately, this is just a very difficult problem to solve. But I'll talk about one other thing that we can do in practice that helps alleviate this problem a little bit. Okay, so now coming back to our original problem, can we use this method to identify motifs that are involved in dendritic localization? So I don't have time to get into the experimental details, but basically we wanted to have a better data set to tell us which RNAs are actually dendritic. So we separated out dendrites and cell bodies and did RNA sequencing on them separately. So that tells us all of the RNAs that are present in those two compartments. And then we can compare them and identify RNAs that have a higher level of expression in the dendrites than we'd expect based on their somatic expression. Then to sort of alleviate the problem of motifs being a smaller part of the region, part of the RNA, we did a sliding window across. So we created multiple sequences from each RNA sequence of smaller windows. And so we put this through our algorithm, and we get about 50 clusters. These are just a few of them. And the most interesting one right now is this one, which was found in 19 different RNAs. And what's really exciting about it is that it seems to be related to a type of transposable element. And that's cool because in the past we've found that transposable elements have been kind of co-opted to create dendritic localization elements. And so we think that this might actually be something very cool. Unfortunately, I don't have any follow-up results yet, but we're hoping to do some experiments on this soon. So with that, I'd just like to thank the Kim lab, especially my advisor, Jin Young, who's always just a huge source of help, and all the other lab members. And of course, I'd really like to thank the CSGF program for this amazing opportunity. So thank you.